Okay. Very meaning afternoon, my dear friends. Today's our resource person, Dr.G. Bhageerath Garu, Assistant Professor, Department of Botany, Government Degree College, Rangasaipet, Warangal District. Sir has giving for valuable time for sharing his huge knowledge and his experiences in plant genetics. So, my dear friends, uh, uh, this is a wonderful platform for sharing and discussing of the important area of the all competitive exams, JL, DL, and CPGET. So better you have to utilize this platform. So we would like to invite today's eminent resource person, Dr. Gogikar Bhageerath Garu. Please welcome, sir, and share your screen and start your session, sir. Please welcome.
Thank you, sir, for giving this opportunity. Now, screen is visible to you?
Yeah, but please click on PDF, sir. It is not opened still now.
Open, sir.
No, sir, no.
Okay, once again, I will share.
No, sir.
Once first I will open that PDF, then I will. Is it visible now, sir?
No, sir. No, sir.
What happened? Now, sir? It is open?
Yes, sir. Yes. Now it is visible, sir. Please, if possible, please zoom it.
Okay. Now it is visible, I think.
Okay, sir. Please proceed.
Good afternoon to all the competitive aspirants. And in the genetics chapter, this is one of the important topic, that is 2-point and 3-point test cross. How this 2-point, 3-point test cross useful to determine the relative distance between the genes. So most of the students are neglected this chapter while they are preparing annual examination or semester examinations. Because genetics is one of the wonderful subject, now we can call it as mathematical botany also. So everybody, student, once you can understood the genetics phenomena or genetic inheritance principles, definitely you can catch the logic in the particular concept. So try to understand. This is one of the important topic. It is mentioned in your PG entrance syllabus also. So 2-point and 3-point test cross and their significance in chromosomal mapping. So you can go for the next slide. Once. Okay. Once recapture, how we can determine where the genes are located and how to map the chromosome. So first we know the frequency or crossing over percentage. We can calculate the frequency or percentage of crossing over in between the two genes. We can use that value as a tool to determine the distance between the genes. We know the percentage of crossing over between the two genes. Definitely we know the distance between the genes. So yesterday class we can know very well that is linkage phenomena is there. This phenomenon was first described by T.H. Morgan in 1910. The genes present on the same chromosome are called as linked genes. You know, every chromosome containing many number of genes. So if A and B genes are present on the same chromosome, they are called as linked genes. For example, here another homologous chromosome is there that is represented as gene 1. In the corresponding another second homologous chromosome represents gene 2. Gene 1 and 2 are non-linked or not linked genes. So you know very well maximum number of genes are present on the chromosome, which always the tendency of the genes is togetherness. They enter into one gamete and they retain the parental characters. That is the concept of linkage. The strength of linkage will be determined by the distance between the two genes. We already know in the yesterday class we also discussed. Are you followed? So linkage can be broken down by crossing over. How to break the linkage between the genes? During the gametogenesis process, meiosis division takes place. At the time of meiosis, in the prophase 1, in the pocketing stage, a crossing over takes place. The exchange of two non-sister chromatids in homologous chromosome pair, this crossing over phenomena break the linked genes to separate the linked genes whenever crossing over takes place at the time of meiosis. Hello, are you followed? Anybody is there? Please give the response.
Sir, yes, sir.
Yes. So crossing over will be more frequent when distance between gene is higher. So in what type of genes generally undergo crossing over? So A and B gene is very close on the chromosome. There is a less frequency to separate these two genes. But A and D gene is there, it is far away when compared to the AB. So that's why there is a more chance, more probability to separate the A gene and D gene. A gene and D gene. So once you can observe here, A and D gene distance is more. That's why if its crossing over takes place at one point on the chromosome, there is a chance A and D linked genes are separate. If the AB gene are very close, that's why there is less frequency to separate these two genes. You can understood this phenomena. Definitely we can go through the 2-point and 3-point test class. Okay, so this is very important phenomena. The linked genes are separated due to the one of the phenomenon takes place, that is crossing over, crossing over process is break down the linked genes. Okay. So just you can observe chromosome maps. What is meant by chromosomal maps? So generally the graphical representation of the genes and the graphical representation of the genes on the chromosome. So which gene is in the first position? Which gene is in the second position? And the order of the genes, and not only that, order and the distance between the two genes, the relative distance between the two genes also we can calculate by using chromosomal maps. First time T.H. Morgan and as well as Sturtevant in 1913, they constructed chromosomal maps related to the Drosophila melanogaster and as well as maize plant. So the relative distance between the two genes we can measure in the Morgan units or map units or centimorgan. What are the units generally used for the measuring the distance between the two genes? That is Morgan units, we can write the answer, or map units, or we can say centimorgans. So how can you calculate the percentage of crossing over? So always we can use the F1 individual cross with recessive parent. That is nothing but test cross. The test cross results give some progeny. Based on the progeny values, we can where the crossing over takes place and what percentage of crossing over in between the two genes, calculate simply, and we can conclude that one. So the crossing over takes place with reference to single point. It's with reference to single point. That is, for example, gene is here and a small a gene is here. Okay. So these two genes are exchanged during the crossing over process. The crossing over takes place with reference to one point on the chromosome, that is called as single crossover. So for example, we can write B gene is here and C gene is here, small b and small c. If the crossing over takes place with reference to 2 points in the chromosome, if 2 points means the complete this segment break and exchange with reference to 2 points, that is nothing but DCO, double crossover. Are you getting my point? Double crossovers and single crossover. Single crossover means with reference to 1 point on the chromosome, the chromatid is breakdown and exchange. With reference to 2 points, that is nothing but 2-point test cross. This is a 3-point test cross, 2-point test cross is also seen in our syllabus. So just you can go through the first problem, chromosomal mapping. The total percentage of crossovers or recombinants of a test cross is if less than 50%, then the percentage can be transformed into linear distance. In CSIR net set examinations mostly ask the crossing over percentage value never exceed 50%. Never exceed 50%. This is very, very important point. The value of crossing over percentage, it never exceed the 50%. So normally, naturally, we can see the crossing over is 1%, 2% or 4%. We can observe in many organisms, but the value never exceed the 50%. You can remember this point. Okay. So next we can go for the continuation of the problem. See here. So this is the, we can consider example Drosophila considering 3 traits with two forms, each to determine the distance in 2-point test cross. 2-point test cross lo manam first oka example chuddam. So this is the dominant features. This is the recessive features. Gray body, the responsible gene is B. Normal wings, that is VG. Normal eyes, CN. So recessive characters are black body, small bg, vestigial wings, small vg, and cinnabar eyes, small cn. So this is the characters and the representative genetic genotypes. See here, we can consider two characters only, capital B, capital VG, and capital VG with recessive type. So this is F1. We are getting F1 is heterozygote. F1 undergo test cross. Test cross is given gametes like this, and the progeny is 41% with the parental characters, with one set of parental characters, another 41% of parental characters with 41%. So what is the distance between the capital B and the capital VG? So how can you calculate? So simply we can write this chromosome. This is B, capital B. This is capital VG. Okay, the homologous chromosome is there, and here small b and a small VG. Okay, so the distance between the two genes is nothing but the recombinant types. How many percentage of recombinant types is here? 9 plus 9. So the percentage in terms of distance between the genes, 9% is nothing but 9 map units. Another 9% is 9 map units. 9 plus 9 is how much percentage? 18. So we can use the formula also. What is that formula? The percentage of crossing over is equal to recombinants by total progeny into 100. The formula is recombinants by total progeny. Total progeny is here 100. So recombinants are 18. So 18 divided by 100 into 100, we are getting 18. So that is percentage. The percentage in terms of distance is 18 centimorgans. 18 centimorgans. So this is the if you assume the chromosome containing 3 genes. What are the 3 genes? B, VG, and CN. So first we calculate distance between the B, VG is 18. Next we can go for another trait. Here B, CN. We can take black body and normalize. Here the recessive characters like cinnabar eyes and gray body and black body. This is gray body, this is black body, this is normalized, this is cinnabar eyes. So here, what is the recombinant percentage? Total is 4.5, 4.5. That is 9%. That is 9 centimorgan distance between the which genes? B and CN. B and CN. Next, we can go for calculate between CN and VG. CN and VG. So CN and VG we can get same 9%. Again, we are getting 9%. So based on these 3 values, we can construct the chromosomal map. This is the chromosome. What are the genes are there? B, CN, and VG. So first we can calculate distance between the B and VG. How much? We are calculated that 18 centimorgan, and B, CN between 9 centimorgan. CN and VG 9 centimorgan. So based on that, the linear order of the genes on the chromosome is B, B after CN, CN after VG. How can you say the CN gene is in the middle? If B and VG, the distance between B and VG is how much? 18 centimorgan. So in the remaining cases, we are getting 9 plus 9. So 9 plus 9 is equal to 18. That's why definitely which gene is in the middle? Which gene is in the middle? CN gene is in the middle. Are you followed? Considering all the 3 cross results, there is only one possible sequence combination which is shown above. So what is the possible gene sequence on the chromosome? We can determine like B, after CN after VG. So this is, we can calculate the distance as well as known the sequence of genes present on the chromosome. So this is nothing but 2-point test cross. We can consider only 2-point test cross, and 2 single crossovers are present here. So 2 single crossovers are there. That's why it's considered as 2-point test cross. Are you followed? Hello? Next, we can go for 3-point test cross. 3-point test cross, we will consider an example of maize plant similar to 2-point test cross. In 3-point test cross, 3 traits were considered having 2 contrasting forms. So we can consider at a time 3 traits, and we can go for the test cross. We are getting progeny like this. We can get progeny like this. The first cross was made between the individual having traits with all 3 dominant forms with an individual having all 3 recessive forms. That is parental plants. From the parental plants, we obtain F1 individual. This F1 individual undergo test cross. The resulting progeny shows different type of combinations. See here. So the respective genes we can identify here. C is the colored dominant. W, capital W is non-voxy, and capital SH full. This is the endosperm. This is the wax coated or non-wax. This is colored. So colored embryo, the wax, presence of wax or not, and shape of the embryo, full or shrunken. So the respective, just you can, I will clear the drawings. So I will draw one chromosome. This is the chromosome, and write the respective genes CW, S. So this is the first parent, and this is the next parent, small c, small w, small s. Okay. So we, from these parents, we obtain F1 individual. F1 individual undergo test cross. We can get the progeny with the different combinations. See here, the F1 individual produced was subjected to test cross, resulting in individuals produced as depicted in table below. So this is the total progeny is 7,000, and we can observe where the double crossovers and where is the single crossovers. That is, you can identify. Definitely, you can calculate the distance between the 2 genes. I will go through the diagram once. It is easy. So the percentage of frequency or crossing over is equal to number of progeny in a phenotypic class by total progeny into 100. That is nothing but number of recombinants divided by total number of progeny into 100. So just you can observe where is the double crossover. That is very important. Double crossovers are identified in the given data. The least number progeny is 4 and 3. The least number progeny is 4 and 3, total 7. These values are indicates double crossover. What is that? Double crossover. So in this double crossover, which gene is in the middle, we can easily identify which gene is in the middle. For example, in the parent CWS and another homologous chromosome containing recessive genes. Here, which gene is changed? C is same as like as parent. W, capital W is also same as like as parent, but small s is there. Small s is nothing but what? Shrunken. Here capital S is there, full. So the S gene is in the middle. Then only we are getting this progeny. So based on the given data in the problem, every student identified least number progeny is considered as double crossover. So double crossover netla identify chestavu. with reference to 2 points. So I will draw this. This is the chromosome. This is 1 point. This is another point. So with reference to 2 points, like this is another homologous chromosomes. So this is 1 point. Okay? This is another point. This is first point. This is the second point. So a segment or a portion of chromatid breakdown and exchange with each other. Which gene is in the middle as per the data? Which gene is in the middle? Anybody say? S gene is in the middle. So that's why we can see here. This S, capital S and small s. So exchange with each other. That's why the combination like this, capital C, small s, capital C, small s. The capital S is exchange with small s with reference to 2 points. So that's why S is migrated to another chromatid. This small s gene is migrated to opposite homologous chromosome. That's why we are getting a different combination. So in this way, you have to identify the double crossovers. So how can you calculate the gene order and distance between the two genes? See here, CSW and CSW. This is the parental type. Middle gene is S. With reference to 2 points, these chromatid portion segment is exchanged and we are getting double crossovers. So based on that, we can calculate that the total recombinants formed between C and S is 1.7, and we can write like this. Formula is also write in this way. Is equal to double crossovers DCO by total progeny into 100. So we are getting total recombinants manak single crossover 1, region 2 and double crossovers. Total is 18.2%. So 18.2% we can write like this. So the 18.2 value. So the distance between the C and S, how much it is? 3.6. So this is percentage in terms of map units. And C and W, how much it is? 18.2%. So 18.2%. So based on that, we can conclude the S gene is in the middle, and CSW, this is the gene order on the chromosome. So this is very, very important in the 3-point test cross and a 2-point test cross. We can calculate the percentage of crossing over. The percentage of crossing over in terms of relative distance between the two genes. So formula everybody remember, every student always. And another one is interference is there. Interference is nothing but in case of double crossovers, chiasmata are formed between the genes. The frequency of double crossovers is found to be much below the expected value. So we can expected double crossover value is equal to the single crossover region 1 and single crossover region 2 into 100. We get the expected value. Actual double crossover value we get from the DCO by total progeny into 100. So the inference is, it is inversely proportional to the percentage of crossing over. Inference is one of the term generally used in the genetical maps. the interference is inversely proportional to the percentage of crossing over. So another interference what says, if the frequency of double crossovers is equal to expected value, there is no interference. There is no interference. For example, double crossovers are not formed in the progeny, that says cent percent of interference is present. Cent percentage of interference is present when double crossovers are not found. If the expected double crossover value and the actual double cross value is same, that indicates that the equal, that is no interference in between the two genes. Another formula is coincidence. Coincidence is actual number of double crossover by expected number of double crossover. So these are the formulas generally used in the construction of genetical maps. That is 3-point test cross and 2-point test crosses are very, very important. For example, in the PG entrance examination, the frequency or percentage of crossing over between the A and B is 20. What is the distance between the AB gene? So we can see simply answer 20 map units or 20 Morgans, like that you can say immediately. So this is the 3-point and 2-point test cross, and how to construct the chromosomal maps by to know about the gene order and distance between the genes and where it is present on the chromosome. So these are the three things we can easily reveal by construction of chromosomal maps, or we can call as genetical maps. So this is the small chapter related to the 3-point and 2-point test cross. If you have any doubts regarding, please ask me. Hello, sir. Hello.
Sir. Okay. So
somewhat it is difficult to the students to construct the chromosomal maps. Most of the students are neglect these chapters because some complexity is there to understand this topic, I think. So yes, by practicing these problems, definitely everybody perfect regard how to construct the chromosomal maps. Okay, sir.
Okay. Regarding of today's class, any doubts, my dear friends? Please ask today's important area for the all competitive exams in genetics area, especially 2-point test cross and 3-point test cross. 2-point test cross and 3-point test cross. This is the most important area for the getting scoring of the marks in JL, DL also. Especially, sir has already previously quoted that without 2-point test cross, 3-point test cross, or construction of the genetical mapping or chromosomal mapping questions, there is no TSET, there is no CSIR net examination also. So, my dear friends, this is very, very important, and a little bit of the confusion and a little bit of the area of the understanding of the subject. So, because of sir has given the very clear-cut idea about the 2-point test cross and 3-point test cross, along with the interference and the chromosomal mapping of the genetical mapping, construction of the gene mapping, the cross or the ratios are most important, and also formulas also most important, my dear friends. So this is the most important class in the plant genetics. So my dear friends, regarding of this class, if you have any doubt, please ask. Okay. So thank you very much, sir. Bhageerath, sir. Really clearly understood what you said in this class. You delivered a wonderful lecture, sir. So tomorrow also same time we will going to meet in this Zoom platform for all competitive exams in plant sciences. Thank you, sir.